We're Group 51 and we chose penicillin for our video topic. We chose penicillin because it's very impactful on society, it saved a lot of lives, and it's something we all really take for granted.
Hi, my name is David Cronk. I'm an IB Sports Science and I worked on the discovery of penicillin in this video.
Hi, I'm Mitchell. I'm an IB Bio and IB Sports Science. I worked on how penicillin works in the body.
Hi, I'm Carissa Stonick. I'm an IB Physics SL and I worked on the modern impacts of penicillin and antibiotic-resistant infections.
Fun fact, did you know the discovery of penicillin was entirely accidental? It all started when bacteriologist Alexander Fleming returned from vacation in the summer of 1928. He was examining colonies of bacteria known as Staphylococcus aureus and noticed a mold known as Penicillium notatum had contaminated the bacteria. Under further investigation, Fleming found that this mold prevented the normal growth of bacteria and could be used to fight infectious diseases. While Fleming did discover the properties of the Penicillium mold, it wasn't until 1940 that scientists would begin to develop penicillin into the life-saving drug we know today. Howard Florey, a scientist at Oxford University, assembled a team of scientists to develop penicillin into a drug. Of his team, Ernst Chain and Edward Abraham found the first method of purifying and concentrating penicillin. The key was to control the pH level of the juice by reducing the sample's temperature and evaporating the product over and over again, essentially freeze-drying it. This process required gallons of mold broth just to produce a fingernail length of penicillin. Andrew Moyer and Mary Hunt alleviated this issue by discovering methods to increase the yield tenfold. Later, Norman Healy came around and made the process more efficient.
Fun fact, have you ever had an infection? Without the discovery of penicillin, you would probably be dead. Penicillin is an antibiotic which only works against gram-positive bacteria, which have thick peptidoglycan layers. Our cells do not have peptidoglycan, making penicillin harmless against the human body. Penicillin prevents peptidoglycan from cross-linking properly in the last stages of bacterial cell wall synthesis. This greatly weakens the cell wall and causes the bacterium to burst open because of osmotic pressure. In this video, you can see on the left penicillin working against bacteria and causing them to burst, and on the right side, it's bacteria growing without the effect of penicillin. As you can see from the chart, there are different types of penicillins which are specialized against different types of bacteria. This is due to the increasing antibiotic resistance from bacteria, and therefore we need specialized penicillin to deal with them.
Fun fact, penicillin-resistant properties of bacteria is quickly evolving. The impacts of penicillin. Risky surgeries. Surgeries such as organ transplants can now be performed with a much lower risk of infection and a higher success rate. It can also be used as a prophylactic antibiotic before surgeries, such as in high-risk surgeries like tooth extractions, which many people used to die from. This is because there are many bacteria in the mouth. Additionally, geriatrics. The life expectancy is much longer, about 30 years, than it was before. Thus, people are living to a much older age, which means an increase in the prevalence of degenerative diseases, such as osteoarthritis, Alzheimer's disease, Parkinson's disease, and cancers. This means that medicine needs more geriatric doctors and more people doing research on the diseases that are the most prevalent among the geriatric population. Now, people are more likely to die of heart disease, cancer, and Alzheimer's disease, or other chronic degenerative diseases rather than infection. Antibiotic-resistant infections are an issue that has arisen from the use of antibiotics. The current antibiotics cause the cell to burst or dissolve the cell membrane, but this breeds for resistance, as the ones that do not get destroyed can replicate. Some examples of antibiotic-resistant infections are methicillin-resistant Staphylococcus aureus, vancomycin-resistant Enterococcus, and Clostridium difficile. There are many more, though. Even the strongest antibiotics currently available, like vancomycin, are not working on these infections. So this means that we need more research in new ways to treat infection. One new idea that has come up is an antibiotic or antimicrobial chemical that blocks the receptor that receives the chemical signal about population size, so that the cells cannot communicate and will not be able to launch a coordinated attack. This image shows how antibiotic-resistant infections come to be. First, an antibiotic is administered, but a few bacteria survive. Then they can replicate, and they can even pass on their drug-resistant genes to other bacteria. This next graph shows how much antibiotic resistant infections have increased over the years. You can see there was a dramatic increase, and it is now a major issue.
In conclusion, penicillin is a world-changing scientific discovery that shaped our history and continues to impact our society today. Just to put the impacts of penicillin into perspective, in World War I, 18% of fallen soldiers died of bacterial pneumonia, whereas in World War II, when penicillin was readily available, less than 1% died of bacterial pneumonia. From its first use to today, penicillin has saved over 200 million lives. So remember, when life gives you mold, make penicillin. Here's the concept map our group made for penicillin. To the right, you can see Alexander Fleming's discovery, connected to other scientists that collaborated to make penicillin into a life-saving drug. To the left, you can see that penicillin caused the cell membrane to lyse, which breeds for resistance because some cells survive. And at the top, you can see how penicillin has directly and indirectly increased life expectancy through performing higher risk surgeries and becoming more successful.